how do we make new drugs? So there are many ways of depicting the drug discovery process. This is quite an interesting concept, a kind of long road, a long journey. Experts in the industry estimate that going from the kind of initial idea of what might be a good drug all the way through to a treatment on the market that would work for patients can take anywhere between 10 and 15 years and cost upwards of $1 billion. So these are the kind of estimates that the industry work on at the moment. And this is one of the things we're certainly trying to improve. At the moment, we're kind of down here at the very early stages of drug discovery, trying to find and make and engineer drug molecules that we can actually then test in our animal models and in patients. Early drug discovery, there are kind of 4 main stages. So there's the basic research that's funded by charities like Parkinson's UK, by the government, trying to understand the basic mechanisms of disease, what's going wrong, why are neurons dying in Parkinson's. That leads to kind of ideas for what we would call a kind of a drug target. So based on the basic research, we identify biological pathways and we think, there's something going wrong here. That could be a good drug target. We'll find a drug that will correct that defect. So we then get ideas for drug targets, and there's quite a lot of effort and work, and a lot of the early basic research really goes into that approach. Then really the drug discovery approach really kicks in at the next stage, where we screen many thousands, tens of thousands perhaps, of small molecule drugs, and I'll explain what I mean later by small molecule drugs. We screen thousands, tens of hundreds of thousands of drugs through some kind of triage process shown by a funnel here, and identify maybe a few drugs that actually hit the target that we've identified. The next stage then is to optimize those hits that come from that initial screen. So quite often there's a conception that those initial hits are the drugs, but they're not. There's a huge amount, as I say, of engineering that is done to those molecules to optimize them really, so that they're suitable, they're potent enough, they're able to get into the brain and access neurons, they're not rapidly metabolized by the body, so they have time to have their effect. And there's basically a cycle of designing new molecules based on the existing molecules you've got, making new molecules using medicinal chemistry, testing those again in your screening systems and seeing how the activity has changed or improved. So what do we mean, first of all, by a drug target? So what are we going after when we're doing drug development? Generally, a target is a protein in a cell. Okay, so the proteins are the kind of molecular machines in a cell that do all the jobs that a cell needs to do, help it move, help it communicate, help neurons to transmit signals. And that protein is normally part of a biological pathway, as I say, that has a kind of an overarching function. And this biological pathway, for some reason, during the disease process, it goes wrong. So this could be a genetic defect that makes this particular biological pathway. It could be environmental toxins or environmental triggers, or a combination of the two that makes this biological pathway function incorrectly. And then we get, as I say, this idea that there is the potential to correct it with a drug. I'm mainly interested in my research in small molecule drug discovery. Okay, so these are the kind of pills that you can take orally every day. And then we move into the phase of drug screening and development to really try and develop a very potent drug that, as I say, can work in a whole animal, in the animal models and in the disease. What is our biological pathway? What are we going after in this virtual biotech, KEAPSTONE Therapeutics? So what I'm showing here is you probably may have seen kind of mitochondria. There's a lot of interest in mitochondria in Parkinson's disease. So this is showing a mitochondrion in the cell. And basically what mitochondria do, they're kind of the powerhouses inside cells. And they take a food source, they oxidize it, they burn it very slowly and in a controlled manner to generate energy, a currency of energy within the cell, something called ATP. So that's energy that's created for all the other biological processes that need to occur inside a cell. This is a normal biological process, but as a byproduct of that, you actually get generated these molecules which are called free radicals. Okay, and these are tiny molecules that are very reactive, and they can bind and damage a whole range of different constituents of a cell and cause damage to the cell. But kind of as life has evolved on Earth, as kind of respiration has evolved, cells have also evolved defenses to try and limit the damage that can be caused by these free radicals. And in one example, the cell defenses might convert them to something harmless like water and oxygen. However, in many neurodegenerative diseases and in Parkinson's disease, this process is not well controlled. And in particular, for example, in Parkinson's disease, if mitochondria don't work well, and there's very good evidence for that, you might generate too many free radicals. So this is just to kind of force this point home, really, that free radicals are a big problem inside a cell if they're not appropriately controlled. So they can bind to all the major different molecules you find inside a cell. So DNA that carries the genetic code, that carries your heritable information, the proteins, the kind of molecular machines within cells, the lipids that envelop the cell and envelop the nucleus of the cell and help neurons to fire and conduct signals, and to mitochondria as well. So free radicals can react with all of these and damage them. And that ultimately can lead to death of the neuron in Parkinson's disease, dopaminergic neurons, and that ultimately leads to Parkinson's. So in many neurodegenerative diseases and in Parkinson's disease, there's an increased free radical production. There are various reasons for this. We don't necessarily understand them all, but there also seems to be these defense systems that I talked about earlier that should mop up these free radicals and prevent them from causing damage. They're reduced. So the overall level of these is much higher in a neuron in Parkinson's than it should be. And that's what eventually will cause all of this damage and lead to neuronal death. So that's our target. So we've done our basic research, and there's a huge body of research that we've conducted and that others have conducted that have implicated mitochondrial dysfunction and free radicals, oxidative stress in neuronal death. Now we need to come up with, we've identified a biological pathway, but we need to come up with, okay, what are the drug targets? What can we actually do with this small molecule drug that I keep talking about? So I'm showing again this biological pathway in the normal system. These free radicals are generated, and there's various cell defenses. I gave one example. There's another cellular defense. This is kind of a regulator of gene expression. It's a regulator of expression of a whole range of different genes. And normally inside the cell, it's bound to another molecule called KEAP1, and that molecule actually stops NRF2 from doing anything. It kind of inhibits NRF2. It prevents its function. And under normal situations, if you get lots of free radicals, they can actually bind to this KEAP1 molecule, they can connect to it, and then they will cause NRF2 to be released. And when NRF2 is released, it can go into the nucleus, it binds to lots and lots of different genes and increases the expression of many different genes, and that has a whole range of different biological consequences, all of which can help to protect the cell from various types of stress, such as inflammation, also this production of free radicals that I've talked about. It can also, quite usefully, protect mitochondria. It can improve mitochondrial health as well. So these are all things that would be very beneficial to do in a disease like Parkinson's. But how can we do that with a small molecule drug? Well, first I need to explain what we mean by small molecule. I need to go into the kind of scale that we're talking about, okay? So we're working at the kind of single molecule scale. I always think of drug discovery as kind of molecular engineering. And we use a unit called the angstrom, this A with a funny zero on top, to measure distances. And 1 angstrom, for the mathematicians amongst you, is 10 to the minus 10 meters, which, for the non-mathematicians, is 1/10 billionth of a meter. Okay, so it's almost a kind of size that's impossible to comprehend. So if you divided a meter by 10 billion, that's the kind of scale we're working at. That's 1 angstrom. So does anybody, any chemists in the room, anyone know what this is? Oh, there we go. No? Very good, yeah. Aspirin, salicylic acid. Okay, so a well-known small molecule drug. Relieve your headaches, relieve fever, kind of reduce your potential to have a heart attack. So this tiny molecule, and I'll tell you how big it is in a minute, can have all of those profound biological effects in the body. And that's really what we're looking for in Parkinson's disease. We're looking for small molecule drugs that can get into neurons and have very profound biological effects. Anybody want to guess how big this molecule is in angstroms? 50? 15, not far off, 6. So that is actually 6/10 billionths of a meter from kind of top to bottom, okay? And that's enough for it to be able to find various targets in the cell and influence them. So what I'm showing you now is the target that I mentioned earlier, this KEAP1 molecule. This is our target. And this is actually, we can use very advanced electron microscopy, this wasn't work that we did, to actually see that molecule, even though it's only 100 angstroms from top to bottom. So this is a protein now, much larger than a small molecule drug. And if we look at that molecule and flip the bottom end here round, that's at the bottom, we can see a little pocket, there's a little hole. And that's where NRF2 binds. So this is showing that pocket in a slightly different representation. There's the pocket in blue and white and red. And the yellow and the pink sticks are different parts of the NRF2 protein, and they fit really snugly into that pocket, and that's why they stick. Well, that's why NRF2 binds to KEAP1. It's because it fits very nicely in that pocket. So that immediately, to a drug developer, brings up the potential to actually find a small molecule drug, something like aspirin, probably a bit bigger than aspirin, that would fit in that pocket instead, and that would actually stop NRF2 binding in the first place. Okay, so we want a small molecule drug to sit in that pocket. And just to emphasize how that would work, so this is the figure you saw before where NRF2 is released and can do all of the good things that it does, and normally that's physiologically with a little bit of oxidative stress will cause that. That pathway maybe isn't happening in neurons for whatever reason. But if we can find this kind of little, this represents a small molecule here, if we can find a small molecule that can block NRF2 binding to KEAP1, that'll have exactly the same consequences. It should release NRF2, get it into the nucleus, and have all of these positive effects in the cell and protect the cell from various stresses. Okay, so we've done our basic research. We've got ideas for a drug target, the KEAP1 pathway. Now we need to come up with some kind of screen. Can we find molecules that actually sit in this pocket? Can we develop a screen for molecules that sit in there? And basically what we did, we had to think about this, and what we did was we took the very specific part of NRF2 that sticks to KEAP1, so this little bit here, you can actually isolate, we can label it, we can put a fluorescent label on it, okay? And we can actually measure that binding to the KEAP1 protein. So we just a little part of NRF2 that normally would bind, and then we can measure that binding, and essentially, when we add a drug that stops binding, this fluorescent molecule changes color effectively. Okay, so with it bound, it's green, with it unbound, it's red, and it'll be unbound if the drug has taken its place. This is an actual screenshot of a computer during one of our screenings. Each square represents a tiny test tube, and what we're looking for are squares that have gone blue. So all of these green ones, they've done nothing, they're inactive. The red ones are really inactive. But there's a couple down here that have gone blue, so those are hits. So you can immediately a visual representation. These molecules change the color of this, so it must be unbound, so therefore these are hits, and we can go and look at those small molecule drugs and see what they are. The problem that we quite often have in academia is that we don't, what you really need to do is screen hundreds of thousands of different potential small molecules, drug molecules. We very rarely have access to those kinds of resources. So actually what we did in Sheffield was we accessed something called the European Lead Factory. And what I'm going to do is show you a little video of exactly what the European Lead Factory is all about. So I think this is really interesting because it's an example of one of the ways in which industry and academia are trying to collaborate to improve drug discovery and make it more efficient and make it faster.
Academics or researchers from SMEs often have awesome ideas for new drugs to cure diseases, but limited ways to develop them. Companies with resources and academics with ideas were often focused on their own field, so nobody ever really joined forces. This was called the innovation gap. That's why the European Commission decided to team up with an association of big European pharmaceutical companies. They invited other companies, SMEs, and academics to join and anonymously share their knowledge and libraries of compounds. This was the start of the European Lead Factory. Simply put, we have a huge library of high-quality compounds that just keeps on growing. Any European researcher from academia or SMEs can submit a target assay or library idea for us to screen. All we need is an innovative target proposal with an assay meeting our technical requirements. A committee of independent and industry professionals will assess the proposal under confidentiality. If they like it, our experts and our screening robot will do their job. After which, you will receive a report of up to 50 hits from our library. Nice! You will open up business opportunities to develop that great new drug and provide patients with the cure they need.
The idea is that ultimately what they want to drive is benefit to patients. So we did this. We screened a library of about 318,000 drugs it was at the time. The other thing they're doing is they're continuously adding to the library, so it's now about 500,000 drugs. And we screened it against that assay that I showed you earlier, and we ended up with maybe about 1,200 that looked interesting. You can get lots of false positives. So when we filtered out things that were giving us false positive signals, we eventually got down to about 20 that potentially looked interesting. And then when we had to actually resynthesize the drugs from scratch, and when we did that, we ended up with about 8 that looked good. And then the European lead factory also made another kind of screening test to really prove that the drugs were binding to our targets. And when we screened through that, we ended up with 2. So from 318,000, we ended up with 2 molecules. So for the kind of biological pathway we're aiming at, that isn't surprising necessarily, but they looked really interesting. So we then did a little bit more work. They did something which I'll explain in a moment. They generated a crystal structure, which is a molecular representation of how the drug fits in that little pocket. And then using that, we can now potentially move on to the next phase. So we've done our basic research. We've got ideas for drugs targets. We've used the European lead factory to screen a big library and find some interesting hits. Now we want to do this bit, and this is really kind of what Arthur alluded to earlier. This is where quite often in academia we hit a problem. So the next phase is what we call the medicinal chemistry phase. So we're going to, we've taken our hits, we're going to design new molecules, we're going to make them, and then we're going to test them, and we need to keep doing that, okay? So how do we design new drugs? Well, what we can do is we can do something called X-ray crystallography. So we can take the bit of KEAP1 that NRF2 binds to, okay? We can add our drug to it, and then we can put it into, say, high salt conditions, and you can make crystals, okay? So just like a salt crystal, but this now is a crystal of a protein bound to a drug. And those crystals, you bombard them with X-rays, and you get a diffraction pattern, and you can use maths to focus that diffraction pattern. It's quite fascinating methodology. You can basically end up with a picture of how the drug sits in the pocket of the protein. So what I'm going to show you now is the actual crystal structure that we obtained. So this is the KEAP1 molecule. Okay, this is part of the KEAP1 molecule. It's like a big barrel. It's going to flip over in a minute and show this pocket on the bottom. Okay, so the blue and the white and the kind of white skeleton structure is the protein itself. This green, red, and blue ball and stick structure is one of our hits, and that's stuck inside that pocket. Okay, and you can basically, using this methodology, this kind of X-ray crystallography, you can really develop a very detailed understanding. Let me just pause it somewhere. A very detailed understanding of how your molecule sits in that pocket and the interactions that it makes. Okay, so this, for example, is a hydrogen bond. So that's the thing that makes water not a gas. Okay, so there's lots of other interactions that are going on here between this molecule and KEAP1 protein. But there are actually also some spaces that we haven't filled. There's potential to make a better molecule that would fit more reasonably in that pocket. So that's what this stage of drug discovery is all about. We design new molecules, we can put this into a computer, we can design new molecules, and we can use the computer to say, okay, can you try and dock this new molecule into this pocket? What's the difference? Does it look better? Does it look worse? Then you can talk to the chemists. Chemists make that drug, and then you test it for real and see if it's made any difference. And you go through cycles and cycles of this until you get drugs that are much more potent, much more active, and also have the kind of characteristics you need in terms of getting into the brain. The problem is that that's very expensive. You need lots of chemists. You need biologists as well running the assays. You need computational and structural biologists to try and understand those interactions and design new molecules. And that costs a lot of money. And kind of, if we look at this graph here, so we've got funding and resources here, and then the stages of drug development. So we've talked about the research stage, the idea, the screening. You know, that can be funded quite well with funding from charities and from government. The later stages are covered quite nicely by kind of funding from the private sector and from drug companies. Okay? And as you can see, in the middle, kind of exactly the stage we're at the moment, the drug optimization phase, there's a bit of a gap. It's hard to get the money to do this. It's too early for a drug company or a private investor to say, yeah, that's really interesting, we'll try and turn that into a real product. And it's a bit late for the kind of typical type of funding that you would get from charities and from government. And that's called the valley of death. So projects die here because they can't be progressed. And that's really where this new strategy from Parkinson's UK is coming in. They're trying to bridge that gap. So they're trying to come in here and use a very kind of applied drug discovery approach, which is quite different from what universities would normally do, to try and do this drug optimization phase. So we set up this company called KEAPSTONE Therapeutics. It's a virtual biotech. It doesn't really employ anyone directly, let's say. It doesn't have infrastructure. It doesn't have an office that you would recognize. All the work is done at an expert drug discovery services company. So the kind of funding from Parkinson's UK comes into the company that immediately then goes to the drug discovery services company as they do the work. This drug discovery company, they've got the right expertise. So we in particular can't do medicinal chemistry really at Sheffield University, certainly not in our department, not in a way that a biotech company would or a pharma company would. But these guys are experts. They've all come from industry. They've been doing it a long time. They have accumulated experience of hundreds of years of doing this very detailed molecular engineering work of turning hits into drugs. So the whole idea is this going to be cheaper, it's going to be faster, it's going to be more flexible. So for example, these kinds of companies are very used to working in a flexible way. If things are going well, you can maybe increase work in one particular area. If they're not going so well in an area, you can move resources away from that area. And that's much more difficult to do through kind of the traditional funding mechanisms. Academics like myself stay involved, so that's great for us. You know, I'm really passionate about this project. I want to see it progress. I want to see it develop. I'm learning a lot about drug discovery as we go, and I hope that we get further with it. There's clarity also for other funders. So this opens up a whole new plethora of potential funders for the project. So instead of the normal kind of grant funding that universities might go to, there's the potential for people like angel investors, venture capital firms to invest quite large amounts. Drug companies themselves could come and collaborate with and or invest in a project like this. And it's very clear who owns what, and that's what these kinds of investors like to see. They like to know what they're investing in. So we keep calling it a virtual company, but I just want to emphasize that there are real people working in this company. So I did want to get a photograph. They seemed a bit camera shy, Signature Discovery. I'm not sure why. Try and get to the bottom of that. But Signature Discovery, we've got a whole group of people working. And as I say, these are all PhD trained scientists who are experts in their field. So we've got multiple medicinal chemists. We've got a computational chemist, the person who can deal with those crystal structures that I showed you and come up with new ideas. And these guys will talk to each other and try and come up with new ideas about ways to move forward, new molecules to make. We actually have as well a crystallographer, someone who's actually making the protein to make the crystals, to generate the structures for the computational chemist to look at. We've got biologists who have taken our assays and are developing those, plus expertise and advice from the senior management team. So the senior management team of this company, you know, they've taken, I think in the last 5 years, they've taken about 7 different drugs from the kind of early stages through to the level of being able to be tested in patients. There's also then a joint steering committee, so I'm on that. Jan Kuligovsky, who's Parkinson's UK drug discovery manager, so he's a medicinal chemist, so he's got a background in the industry, so can input to the project and really keep an eye on these chemists. I'm okay on the biology, but I'm not so great on the chemistry, so it's great to have someone like Jan. But also Alan Naylor, so he's ex-head of chemistry at GSK, again, very creative scientists who can come up with new ways of doing things, new ways of working, and have been there and done it, you know, have taken drugs all the way through. And then we also have Tom Bartlett, who's working more on the kind of business development side. He's at Sheffield University. And then finally, we've got the board, the actual KEAPSTONE Therapeutics, the board of the company itself. So Steve is on that board, Arthur as well, myself, and then 2 more people from the university who mainly deal with the finances and the kind of more business development side of things. And we all interact. There are very set ways in how we interact with each other and the kind of information we pass to each other and how we work together. But most of the time, it's the Joint Steering Committee interacting with the contract research organisation on a weekly basis. Very often we meet very regularly to see what results we're getting and to try and drive the project forward. So what are we doing? Well, it's this, as I said, design, make, test cycle. And there's a huge amount of work in this, and so far what we've done, we're, as I say, at very early stages. We've set up and established the testing methods, or Signature Discovery have. They've developed some new testing methods, actually, which add a huge amount of value to the project and open up other avenues for trying to find new molecules that can hit this target. We're making lots of new drugs and testing them in the assays. We've actually made some more KEAP protein for crystallography to generate the crystal structures. And we are still, as I say, at the early stages. The goal is to make drugs that fit better in the KEAP1 pocket. So the drugs we've got at the moment, they stick in maybe like as if they were put in there with Pritt stick. We need them to be super glued in. So that's really the focus at the moment, is to try and make them bind much more strongly, at the same time making sure they have the potential to get into the brain.